Good afternoon, everyone. I am Lucrezia. I'm a postdoc in the UCL Cancer Institute with Simone Zaccaria. And today I am presenting POPSICLE, which is my probabilistic method to infer copy number probabilities in single cells. And so cancer cells are known to acquire throughout the evolution of the disease distinct type of genetic alterations, and a common type of alterations are the copy number alterations that result in the amplification or the loss of either large or short genomic regions, such as the one presented here in this slide. And these alterations confer a selective advantage to cancer cells in cases when amplifications, for example, hit relevant oncogenes or when losses hit relevant tumor suppressor genes. And these alterations can be acquired individually by each cancer cell. And so if we are interested in understanding all and analyzing all the alterations that have been accumulated in each cancer cell, we want to exploit single cell DNA sequencing technologies, such as, for example, DLP+, that allow us to isolate each single cell and then extract a signal that is a read count signal that is such as the one presented here in the slide, where we have that this signal is proportional to the copy number. And so we can see that we can use higher read counts to infer amplifications, and we can use lower read counts to infer losses. And basically, over the years, using this idea, many computational methods have been proposed in order to group each genomic bin into segments characterized by the same copy number that here in this slide is represented in different colors. And then these methods use these copy number values to then infer the classes of events such as losses, gains, and amplifications. However, this inference is still error-prone for existing methods, because when we are trying to infer the exact point estimates, there can be cases, such as in this region that I'm highlighting right now, where we might have false negative events. Where, for example, here in this slide, I am showing that the purple bins that are with an amplification event have been inferred with the wrong lower copy number value. And so in this case, we are missing this amplification. Or, for example, if we focus on another genomic region, we can see that in this case, we have these bins, these red bins, that have been inferred with the wrong higher copy number value. And so in this case, we have a false positive event. And the reason why this is happening is that read counts are very dispersed. And so in certain genomic regions. So, for example, if we have a look at this genomic region where the signal is not very dispersed, we can see that in this case, the inference of the exact copy number point estimate is very easy, because there is only one copy number state that can explain our data. However, when we look at other genomic regions where the dispersion is much higher, we can see that now there are multiple possible copy number states that can explain our data. And so the inference in this case of one exact point estimate is more difficult, because in these cases, so in these genomic regions with high dispersion, this dispersion makes it unfeasible to correct the exact point estimates. And so the key idea that we are proposing now is, instead of looking at the exact point estimates of copy numbers, we want to have a method that allows us to look at the probabilities of having different copy numbers in each genomic segment. And in practice, what does it mean? This means that if we have a look at the region where the inference was easy, the copy number probabilities would tell us that there is only one possible copy number. Instead, if we now look at one region where the inference was more difficult, now having the copy number probabilities allows us to know that our point estimate might be incorrect, and tells us that there are actually, in this case, 4 possible copy numbers between 8 and 11 that can explain our data. And so, with this idea, we have developed POPSICLE, which is a framework that in the end provides as an output the copy number probabilities for each genomic segment. So our method is divided into 3 main steps. In step 1, we perform a first round of segmentation of the cell, and we identify the segment that can correspond to the lowest copy number of the cell. And we use then this as a reference copy number to compute a set of different ploidy candidates for the cell. Then for each of these ploidy candidates, we perform steps 2 and 3, where we compute the copy number point estimates and we obtain the segmentation of the cell. And then for each of these segments, we then compute the probabilities. And we repeat these steps 2 and 3 for each of our ploidy candidates, and then we run model selection to then choose the ploidy candidate that best explains our data. So after we have implemented POPSICLE, we have developed a simulation framework to assess its performance. And we made this simulation realistic by downloading ground truth copy number profiles from high quality previously published cells. And then we have used these ground truth profiles to generate 1,000 single cell read counts profiles by varying two main parameters that are the sequencing coverage and the dispersion. And here in this slide, I am presenting two single cell read counts profiles that have the same copy number, ground truth copy number profile, but on the left we have the highest possible complexity in our simulations, that corresponds to having low coverage and high dispersion. And then on the right instead we have the least complex, which corresponds to when we have high coverage and low dispersion. So then we took these generated read counts profiles, and we have assessed the performance of POPSICLE in, first of all, performing what the state-of-the-art tools perform, which is inferring the ploidy, inferring the segments, and inferring the copy number point estimates. So in terms of ploidy inference, we can see that if we compare our performance to a commonly used method in the state-of-the-art, which is HMM copy, we are able to improve the inference of the ploidy, both in cases where we have low dispersion and high dispersion. And then when we consider instead the breakpoints identification, we can see that also in this case, we are able to improve the accuracy in the identification of copy number breakpoints. And when I say breakpoint, I mean a position in the genome where we have a change in copy number. So overall, we were able to observe that in terms of ploidy inference and breakpoint identification, POPSICLE has returned better results compared to the state-of-the-art. And so now, after having assessed POPSICLE performance, we then applied it as a proof of concept to 1,700 triple-negative breast cancer cells. And in the remaining part of the presentation, I want to show you two possible applications of our method, starting from how we can use our method and our probabilities to improve the copy number event detection. So here in this slide, I am showing the copy number point estimates. So the copy number values that best explain the data. And on the x-axis, we have the cells, and on the y-axis, we have the genomic positions. However, the main output of POPSICLE are the copy number probabilities. So now, if we have a look at this heat map, we are representing in magenta the regions of the genome in each cell that are affected by a gain, with increasing thresholds on the probabilities returned by POPSICLE. And then if we summarize this information across the cells, we obtain this frequency plot where we have on the y-axis the fraction of cells that are affected by a gain in each genomic position for increasing thresholds on the copy number probabilities. And so you can see that there are regions such as, for example, chromosome 8, where the frequency of amplifications remains the same also when we increase the threshold on the probabilities. Instead, we have other regions such as chromosome 9, for example, where the frequency of gains instead quickly drops as we increase the threshold on the probabilities, meaning that the model is not confident enough to call this region maybe as a actual gain. But now still the question remains as to how are these probabilities, what do these probabilities give us that is different from what the state-of-the-art gives us with the copy number point estimates. And so now if we focus on amplifications and we report the frequency of amplifications across the genome inferred by HMM copy, and then the frequency of amplifications inferred by POPSICLE on the bottom, we can start to compare the results. And for example, if we focus on chromosome 17, we can see that there is this genomic bin, for example, where HMM copy has not inferred an amplification in basically any of the cells, and instead POPSICLE has identified an amplification in nearly 80% of the cells. And And this is a bin that contains the gene SOX9, which is a gene that is known to be frequently amplified in triple negative breast cancer. And now, if we have a look here at the read count distribution of in one of the cells in this dataset, we can see that the bin that contains SOX9 has a higher read counts compared to the neighboring bins. So this is an example of a method that might have been missed by using a state-of-the-art approach that only looks at the copy number point estimates. And now, on the opposite, we can have a look at chromosome 8, where we have, as I said, the opposite case. So we have a genomic bin where HMM copy has inferred an amplification for basically 100% of the cells, and instead we did not identify it, so this one here. And this is a genomic bin that contains EXT1, which is a tumor suppressor gene annotated in COSMIC. And you can see that here I am presenting the uncertainty returned by POPSICLE, and you can see how these genomic bins are characterized by high uncertainty, but their probability of being amplified is much lower when we compare it to their probability of being gained. And finally, if we have a look again at the read count, we can see that these genomic bins that contain EXT1 have lower read counts compared to their neighboring bins. And so now, after I have shown you how we can use it to improve the detection of copy numbers, we can then now, I can show you how we can identify regions that might have the copy number solution uncertain. So what we did was we wanted to investigate whether there was any relation between the position of the bins with respect to copy number breakpoints. And you can see that there are, here is a read count profile, and you can see that there are some genomic regions where the density of breakpoints is lower, and then there are other genomic regions that are characterized by a higher amount of breakpoints. And here the read counts profiles are colored based on the copy number point estimates. And if we now have a look at the probability of this copy number point estimate solution, we can see that the regions that are farther away from the breakpoints have a much higher probability, higher than 0.8, and instead the genomic bins that are closer to the breakpoints have a lower probability for their corresponding point estimate. And now if we extend this solution to all the cells in our dataset, we can see that overall we have observed this trend where we have that the breakpoints that are closer to a breakpoint are more uncertain, here in red, and then instead the breakpoints, sorry, the bins that are farther away from the breakpoints are characterized by lower uncertainty. And so this concludes my presentation, and I have shown how, how point estimates of copy numbers can be insufficient to correctly identify all the gains, losses, and amplifications. And we have presented POPSICLE, which is a method that infers copy number probabilities in single cells. And I have shown how we can use POPSICLE to better characterize copy number alterations, and how we can identify bins where the corresponding copy number point estimates can be difficult. And I would like to thank you all for your attention, and especially my lab for the constant support in this work. Thank you very much. And I'm happy to answer any question.
Thank you. All right. Do we have any questions? Yes. One question is coming.
Hi. Really great work, really cool tool. I just wonder, because the world of single-cell whole genome sequencing is not characterized only by DLP+, I just wonder whether your method would work on something which allows more coverage, but more uncertainty as well, like PTA, for instance. Do you need to tweak it somehow to work with another method?
So based on the assumptions of our model, we can also work with higher coverage data. And also that's why I've also performed some simulations with higher coverage to assess the robustness of the method. Also with in cases with higher coverage, we are still able to compute the copy number probabilities.
Great. And also, just a short question, do you address somehow the copy neutral loss of heterozygosity in this method, or just?
Yes, so our method, I didn't comment on that, but we infer allele specific copy numbers. So the likelihood of our model includes both the read counts and the SNP counts. So we can infer also copy neutral LOH.
Cool, thank you.
Thank you.
Yes, another question.
Thank you very much. A kind of a mouthful, the spelling out of POPSICLE. It's wow. Good job.
Thank you.
And I have a question that is more, it's unrelated to single cell, but I think this is like perfect to identify like difference in ploidy, like for when we suspect that something has a wall genome duplication and should be. So my question here is like, for your probability, I think you need numbers of events that you see. So in this case, is the cells, the individual cells. Your breakpoint analysis could be used without the many cells. Like if I just have coverage, like over a genome,
Yes.
then should be able to spot the.
Yes, so our model works with each cell independently. So we can also work, it also works if you only have one read count profile, if that was your question.
That was me.
Okay.
Grazie.
Thank you.
More questions? I have one. So what about rare cell types? Will you be able to detect also the copy numbers variation when you have a very, very rare cell type?
Yes, because since our model works with each cell independently, then we don't discard any copy number alteration. And then you can use the output with the probabilities and the point estimates to then decide at which granularity you want to cut your clustering, and then I study more in depth, like more rare alterations.
Cool. Any other questions? All right.
Thank
Thank you
you
so much.
Thank you.